So chapter 6, enzymes. Enzymes by and large are proteins. We can have some enzymes that consist of RNA, we call them ribozymes, but most are going to be proteins. And enzymes are said to be this big fancy word, catalysts for biological reactions. Catalysts don't make reactions happen. Catalysts facilitate reactions in the sense of they can make them happen faster, and they can help them happen without using so much energy. So our illustration here, this would be a spontaneous reaction if we look at our free energy. And of course, our reactants have a higher energy than our products. So what is our delta G going to look like for that? Positive or negative? Negative delta G. That's going to be a reaction to the right, so that's a spontaneous reaction. But notice we've got this bump right here in the middle. That is referred to as our activation energy. And so this is the part that often for spontaneous reactions to occur, that's the difficult part, going up the hill before we can go down the hill. I like to think of this like a roller coaster. You like to ride roller coasters? What's the very first thing you go, when you get in the thing and it starts moving, what's the first thing that's going to happen? Cha-chuck, cha-chuck, cha-chuck. You got to go up that big hill, right? Building up that potential energy. That's the activation energy. Okay? And that's normally what's going to happen. So when we look at our free energy, especially an equation like this, can it go? Will it go? It may. And in this case, we see it spontaneous and we go, yeah, it can, if we can get over our activation energy hump. Well, enzymes, these catalysts that facilitate these reactions, in fact, can act to lower the activation energy. In this particular reaction where we're taking ATP with water and an enzyme, we're going to produce ADP and phosphate. And so lowering that activation energy, let's use our analogy of a roller coaster. Have you ever been on a roller coaster that didn't send you up that big hill first? Yeah. It's like you get thrown off an aircraft carrier, right? Have you been in those things? Oh, they push you against the seat because this catapult has shot you out of the starting gate. That's an enzyme. It's providing that energy up front rather than having to go up that hill and you're at top speed a lot faster. That's the enzyme, adding the speed. And I like my idea of the roller coaster because those roller coasters without the hill have a catapult to make you go fast. Enzymes are catalysts. See how it sounds the same? See what I did there? That's an enzyme, to make you go faster without the big hill. So the catalyst increases the rate. You don't have to go up the hill. You're going faster sooner. And what we're going to see that enzymes are able to do, they're able to reach this transient form. We're going to see enzymes are not these big metal inflexible wrenches. Enzymes are extremely flexible. They're proteins, so they're going to be able to change shape. And enzymes are actually going to grab onto the reactants. And they are going to, as the enzyme changes shape, they're going to change the shape of the reactant, or in this case we call them substrates, such that it's easier to process. And changing the shape of that substrate is referred to as a transient state, this transient complex. It's only temporary, so we can process the substrate, and we think of enzymes cutting a lot, so you'll see pictures of Pac-Man as you talk about enzymes. So enzymes can help form these shapes and these bends, conformational changes of the enzyme itself and the substrate that will allow the reaction to go faster and easier using less energy. And by doing these things, you are absolutely going to alter that equilibrium that we spent all last chapter talking about, because the enzymes are going to favor in one direction that at a natural, normal state in the environment would be at equilibrium.
Can you repeat what the word transient means?
So you start off with an enzyme and you got a substrate. So I think in some ways this is like cutting out snowflakes. You ever cut out snowflakes out of a piece of paper? You got your scissors and you got a piece of paper. You fold that paper several times over, right? That's a transient state because it's not going to stay like that. The scissors start cutting that folded up piece of paper and it's still folded even after you cut. That's a different transient state. And then when you separate the scissors from you unfold it, now you have the final product. Enzymes are going to take that.
Something that's constantly changing?
Well, it's temporarily changing. Okay? Your transient state is going to be different from the reactant and it's different from the product. It's just the intermediate. It's the intermediate in chemistry. You know the intermediates of a reaction? Transient state's like an intermediate. And it's relatively short-lived, and so that's why they say transient. Yeah. So there's our enzyme. Thanks to the amoeba sisters. So we have our enzyme. Our enzyme is going to physically interact with a substrate. And that location of the enzyme in which the substrate can interact or bind with is referred to as the active site of the enzyme. Now, this binding is usually going to be facilitated by interaction between the R groups of amino acids. And since this interaction is temporary, what kind of bonds do you think are going to form between the substrate and the enzyme? Hydrogen bonds. Why?
Because it's the weakest.
The weakest bonds. You can interact, but you can also separate. You would not want a substrate binding to an enzyme with a covalent bond. Why? You just super glue them together. They would never come off. And as you look at one specific enzyme, what they have colored in blue and green and pink and red are what are referred to as prosthetic groups. And these prosthetic groups are extremely important because you see this is the pocket, this is the active site within this particular enzyme. And only a very specific substrate is going to fit. And when that right substrate fits, these groups that line the active site that are critical for the activity of the enzyme are going to grab onto the substrate and lead to a change of the shape of the enzyme and a change of the shape of the substrate into this transitional, transient, intermediate processing form. And that's largely how enzymes and substrates work. So that active site is going to be very specific for what material it's going to process. Enzymes aren't generic in a sense. Some will say, depending on where they bind, because we have proteases that can bind proteins, we will see some of those just chop off amino acids at the end because they're looking for the peptide bond. And where are you going to find a peptide bond in a protein chain between which amino acids? Which specific amino acids are going to have peptide bonds? That's a trick question, by the way. All of them. They all have the peptide bond regardless of which amino acid it is. So, now just a little bit of understanding. As you look at enzymes down the road, if you're working in a lab and you're using enzymes for biochemical experiments, you're going to see that their nomenclature has changed. I'm old school. And I'm old school not just because I'm old. I'm old school because there was a reason that we did things and it made sense. People come along and try to make things better, and invariably it doesn't always work out that way. If it's not broken, don't fix it. You ever heard that one before? So when we look at the nomenclature of enzymes, they used to be named based on their substrate and the function. I just said proteases, right? If you hear ASE, you know it's an enzyme. And typically the first part of that name tells you what's being processed. So a protease processes what? Protein. Guess what a lipase processes? Lipid. So in this sense, a nuclease, what does that process? Nucleic acid. That makes too much sense, right? Yeah, that's too easy. Let's change it. Let's all of a sudden start sticking numbers on these things. And so this International Union of Biochemistry, a committee, right? Anytime you see a commissioner or committee, you should get very nervous. Because typically it's not going to work out well. Look at what this group did. They said, okay, we're going to have 6 classes of enzymes. And so they labeled enzymes EC. There's class 1, class 2, 3, 4, 5, 6. So instead of having an enzyme that we call alcohol dehydrogenase, that name makes sense, right? This is an enzyme that's going to have this dihydroxy group on. It's going to take it off. You know in the name of the enzyme what it does. Now that enzyme is called EC1111. So you have to either memorize EC1111 and know that it's alcohol dehydrogenase, or you got to go look it up. So understand that the descriptions of enzymes usually include both simply because the old school is better. It makes more sense. I'm going to go old school. I'm never just going to throw out numbers for you. All right? Now that again is just sort of for your benefit to understand in the future if you're reading research articles, if you're working in a lab, if you're in a class where someone goes with the very modern EC numbers, you know when you see those 4 numbers, it's talking about an enzyme somewhere and you got to go Google it to find out which one it is. All right? And you'll find typically in the definition, you'll find the old nomenclature, which is the name. It usually tells you what it does. Now, like most anything in nature, your body, your physiology, how you feel is going to be affected by temperature and pH. Have you figured that out yet? I get cranky when I get hot. I don't like being hot. You can put me in an ice cold room and I'm going to be, I may not be comfortable, but I'm not going to be cranky. But if I get hot, I get cranky. Likewise, if you in your diet, you mentioned the Atkins diet before, if you take that to the extreme degree, which I did a couple of decades ago, almost like an experiment on myself or just being stupid, you figure it out. Atkins diet is a low carb diet. I decided, well, if low carb is good, no carb probably even better. Lose weight faster. Have you ever had people that do that? Yeah. Okay, so I had no carbs at all. I only ate protein for several days. Well, when that happens, your body starts, once it burns through your carbohydrate stores, and it really starts to burn through your fat stores, and this was when I was in graduate school and about the size of a piece of spaghetti, so I don't know why I was doing this in the first place. It seems like the skinniest people are the ones that think they're the fattest. Figure that one out. Anyway, your body starts burning protein instead of carbs and lipids, and when you burn protein, you produce these things called ketone bodies, which starts to make your blood more acidic. And you go into what's called ketoacidosis. And I remember sitting at my desk, I was getting lightheaded. I had no energy, and I'm like, what in the world is going on? Blood sugar and pH changes. So someone had a Snickers and they said, here, eat this. I love Snickers, by the way. I plowed that thing down. In about 10 minutes, I was feeling great. So when your pH changes and your blood pH is 7.4, if you go below 7.35, you have acidosis. If you go above 0.45, you have alkalosis. Your body does not like any changes in the pH because enzymes are affected by that. And when we look at this particular graphic, we're going to see that a typical human enzyme, its optimal temperature is going to be working right around 36, 37 degrees centigrade. What is normal body temperature? 36, 37 degrees centigrade, in that neighborhood. 98.6-ish. But when we look at an enzyme that's found in a bacteria that lives in a hot spring, all of a sudden, look at its optimal temperature. 100 is boiling. I cannot imagine living in that. But enzymes are optimized for those living conditions. So that's our temperature. Here, pH. We have an enzyme trypsin that's a protease. We have a different enzyme pepsin. It is a different protease. It works differently. Here, optimum pH for trypsin is around, I don't know, maybe a little bit above 7.5, but it's closer to 7.4. Do you see that? That would be about right for human enzyme. But here we have this enzyme pepsin, and its optimal pH is closer to 2. What organism do you think produces and uses this enzyme pepsin? Some cold-blooded animals do, but I will give you a little hint. Pepsin is an enzyme found in the human body. Stomach. Why? What do you have in the stomach? Hydrochloric acid. Pepsin is a stomach-produced enzyme to help break down proteins in the stomach under acidic conditions. Trypsin is going to come into your digestive tract in the first part of your small intestine, where it's injected in from the pancreas. It's produced in the pancreas. And some from the liver. But by the time that material gets into your small intestine, you've produced a ton of sodium bicarb and neutralized the acid, and your pH is back in the neighborhood of 7. So enzymes are optimized. Protein structure is optimized for temperature and for pH. All right, so now, enzyme substrate. Enzyme is the factory, enzyme is the worker, your substrate is your building material. And you can think of your substrate as a lump of clay and the product, the finished vase or statue, or however you want to look at it like that. So there are two models, essentially, of how enzymes work. The first is a classical model that really gets at substrate specificity. And it's called the lock and key model. How many of you have a car key? Will your car key open and start any car in the parking lot? It's specific for your car. The lock of your car is the enzyme. Your key is the substrate. Do you see? Lock and key. So that gets at our substrate specificity that we see here. There's the lock, there's the key, fits. But then all of a sudden there's like this black box. Something happens. Poof. And then we end up with our product. So as we begin to know more and more about how proteins work, about the structure, primary, secondary, tertiary, quaternary structural proteins, we realize that proteins aren't that lock of your car, that inflexible piece of metal. They can change shape, change conformation. So in almost 1960, the induced fit flavor of how enzymes and substrates interact came into play. We still have our substrate specificity, our active sites, in which our substrate binds. But what happens to the substrate and the enzyme after they bind? What have both done? They've changed shape. They've changed their conformation. That is a transient state, a transitional state, an intermediate state. And do you see what's happened to our substrate there? I mean, it would be difficult to cut through that whole middle of that substrate, how thick it is. But when the enzyme bound that substrate, it pulled it apart, so there's just that little bit left that can be cut. That's an illustration to get to the point. That now, because of that interaction, that's a whole lot easier reaction to occur. One little snip, you release and reset, and you get your products from your substrate. You see the difference between the lock and key and the induced fit? Induced fit, only true difference is the conformational change, and that's what lowers the activation energy to then allow the processing of the substrate into your final release products. So here's our amoeba sisters again. Now, the only thing that I wish the amoeba sisters had done is make our substrate change shape as well. You can see how the enzyme is hugging and getting close after it binds to the substrate. But again, substrate-enzyme interaction, what bonds typically? Hydrogen. You can have some ionic bonds, but hydrogens are much more common. And again, the key to the induced fit is the change of the shape. So, with induced fit in mind, let's look at this example. These are the steps. And again, we're using a specific one, but this is the generic. This is how enzyme substrate works. First, substrate's got to bind. How does the substrate and the enzyme get together? How does the enzyme and how do they find each other? Match.com? I mean, what'd you say? Random collisions of molecules just floating around and spinning around. All of a sudden, the substrate that's the right fit, in our case, sucrose, bouncing around in here, boom, it hits the active site. Once the substrate gets in the active site, we're going to activate the substrate, which also activates the enzyme to change our shape. This is where we get our transient form. Substrate activation, transient form. I wish they had put another one in here, because in between our activation and our product release, we're going to process. So this is going to be transient form processing of our substrate. Then the product is released. You can see the release here. For our enzyme sucrase, takes in a disaccharide. It's going to release 2 monosaccharides. And then the enzyme has to change shape back to the starting position so it can do what? Collect another, start over. So there's our cycle. So you bind the substrate, activate, process, transient form, release the product, reset, and do the show all over again. So that's generic for just about any enzyme you could plug in there. Yeah.
Can you repeat one more time what's in the substrate?
So this one, I mean, this is I think it's too general. Substrate activation, this is transient form, which is the changing of the shape of the enzyme and the substrate. But this is also where you process the substrate. And you convert the substrate into the product. Because you can't release the product if you haven't made the product. So do you see how that's a kind of a very general term, but lots happening right in that step number 2.
Okay, this is just what I got from what you said earlier, but I said that whenever it hits together, that makes the enzyme change shape, and then when that changes shape, it'll change the shape of the substrate.
Correct, and that's all happening right there.
Yeah.
But what gets together the substrate binders is just random chance. A lot of stuff is hitting in there and they're moving really fast, but only the right fit, the right key is going to stick and cause this next step. Now this is another one of those things, sort of like bioenergetics, not my favorite topic in the world. Enzyme kinetics is what this is usually talked about in biochemistry. Enzyme kinetics. When you look at the concentration of your enzymes and substrates, certainly the more you have, the better the chance of having a random match and a processing of your substrate. So as your concentration increases, and this is substrate concentration, you're going to increase the rate at which the enzyme is processing. Makes sense, right? Because typically enzymes are going to work faster than you have enough stuff to work on. And you can see we've got pretty much an exponential, just upward line, almost a straight line. But what's starting to happen here?
It's slow. It's still going up. But it's
Why is it slowing down? It reaches the maximum. And what's happening when you get up here at the max? You got more stuff to process than you can process. This takes me back to the old I Love Lucy show. Have you ever seen those reruns, I Love Lucy? There was a episode where she and Ethel were working in a candy factory on the assembly line. Classic. You remember that one? And they kept hitting the button that made the conveyor belt go faster and faster and faster and faster. When the candy was first coming, it was easy. But then it got faster, and they didn't know what to do with the candy. She started putting it in her pocket and eating it. Now, that's what's happening. You've got so much substrate, the enzymes are like, wait, stop with too much. And so you reach your maximum velocity with which those enzymes can process substrate. And it plateaus because the enzymes are like, look, you can keep sending the stuff, but we can't process anymore. And to help get an idea of enzyme velocity, enzyme kinetics, how fast can enzymes process substrate, there is a measure that is called the Michaelis-Menten constant, the Km for an enzyme. And that Km is going to be at half the maximum velocity. See, there's our max, so we're right in the middle. If you take that half max velocity and find the concentration of substrate required to reach that half max, that then is the Michaelis-Menten kinetics constant. And you will always find a Km for any enzyme that's listed in that EC guide. It tells you how efficient, how fast enzymes can actually work.